Okay, so hello everyone. My name is Helena Cornu. I'm the outreach officer for Open Targets, and I'm here today to talk to you about the Open Targets platform and how it can help you identify and prioritize drug targets. So how I thought this webinar would go. First of all, I'm going to talk to you about Open Targets, the consortium, to give you some context and to give you an introduction to our vision and how our informatics tools and informatics ecosystem fits within that broader vision for drug discovery. I'll then move on to the main topic of the webinar, the Open Targets platform. And this is particularly exciting because just last month, we released the new next generation version of the platform with a whole new design, a suite of amazing new features, and the code base has been entirely rewritten. So it's much more efficient and much more performant. So I'm really excited to be able to tell you about it today. I'll then talk to you through some use cases and research questions to give you an idea of how you can use the platform in your research. And then also I'll give you some tips on how you can best access the data and what are the best ways to use that data depending on how complex your query is. And then finally, I'll just summarize it. And as I said, there'll be a Q&A, so you can pop any questions you have in the chat and we'll talk about them then. So, to start off, we'll talk a little bit about open targets. As many of you know, the reality of developing a new drug is that it's a really lengthy process. It's a costly process with low success rates and high attrition rates. This graphic gives you an idea of just how difficult it is to get a new drug approved and how much failure there is along the way. Many drugs will fail this process, usually because of a lack of safety or lack of efficacy. And ultimately, I think we can all agree it's a waste of time and resources. So what can we do to improve this? Well, the first step in the drug discovery process is to identify a target. Now, this could be a gene, a protein, or another kind of molecule. And that target, if we modulate it, we hypothesize that it'll benefit patients in disease. Once we have a target in mind, we can then think about creating and testing drug candidates that will perform that modulation selectively. Of course, there are other considerations. For example, there has to be a clinical need for it, there has to be a commercial need for it, but really the most important step is choosing that target because that's what kickstarts the whole process. And as I mentioned, many targets fail the clinical trial process due to a lack of safety or lack of efficacy. And what this tells us is that often our understanding of our chosen target's underlying biology is incomplete or just wrong. The graph which I show here is from a paper by Matt Nelson, in which they looked at the success rates of different compounds throughout the clinical trial process, and they found that compounds are much more likely to progress through the pipeline when they have direct genetic support. So basically, the more information we have about a target to start off with, the better chance we have of creating a successful compound to modulate it. And so this is where Open Targets comes from. So Open Targets was founded to address this challenge by integrating public data that provides evidence to associate a target with a specific disease, and by generating experimental data that supports causal associations between targets and diseases, and by generating foundational understanding of disease biology. So Open Targets is an innovative, large-scale, multi-year, pre-competitive industry-academia partnership, and we use human genetics and genomics to systematically identify and prioritize targets. So don't panic, I know that's a lot of buzzwords, but in the next few slides, I'm going to go through all this and by the end, you'll understand exactly what Open Targets is about. So the first thing you need to know is that it's an industry academia partnership. So this means that we have a number of collaborators. So at the heart of what we do are 5 complementary institutions that bring their respective expertise to the table across academia and industry. So we have 2 academic partners. These are the EMBL-EBI and the Wellcome Sanger Institute, both based at the Wellcome Genome campus. EMBL-EBI brings their expertise in life science data, and they have a range of data resources that we can integrate into our informatics tools. And the Sanger Institute brings their expertise in genomics and their amazing experimental capabilities and labs. Beyond that, we currently have 3 industry partners. These are GSK, BMS, and Sanofi, and they bring the industry perspective. So what the pharmaceutical industry is looking for in terms of target identification and drug discovery, and they also bring their expertise in their respective therapeutic areas. Together, all 5 of these institutions bring very complementary strengths that enable our consortium to pursue and fund experimental and informatics research. So we have 2 broad categories of research programs. The first is the experimental research program, so the lab-based research program. And this uses a number of high throughput methods to generate target-centric data in a variety of human systems. And in this program, we focus on 3 therapeutic areas. These are oncology, immunology and inflammation, and neurodegeneration. Alongside this, we have our informatics program, which looks at using the breadth of publicly available data to help users sift through all that information and identify the targets that will be most useful to them in their research. So we know, because our team works with this on a daily basis, that there is a huge amount of data out there, and one individual will have a really hard time to sift through all that and find exactly what they need to find novel associations or novel targets. So our aim here is really to make this as straightforward as possible and as comprehensive as possible so that we don't miss anything. And to give you an idea of our approach, this is a schematic of our knowledge cycle. So our informatics programs take publicly available data, integrate it in order for researchers to be able to generate therapeutic hypotheses. Our experimental research program carries on from that, takes those hypotheses to the bench, generates experimental data, and then makes that data publicly available, ready to feed straight back into the platforms. And I should point out here that our informatics tools are all open source and our data is all open access. And that commitment to open sharing is really something that we value at Open Targets. So broadly speaking, that's what Open Targets, the consortium, does. I'm now going to focus on the Open Targets platform, which is our flagship informatics product. And as I mentioned, as of a month ago, we have a brand new, redesigned, rewritten platform with a completely new code base, new data, new features, and it's really powerful and really cool. So you can access the platform on the web. So the URL is platform.opentargets.org. And the platform is essentially a data integration and analysis tool. We integrate over 20 different public data sources, which we use to build and score associations between targets and diseases. If you use the web interface, we support searches by target, diseases, or drugs, but we also have another number of ways in which you can access the data, including an API and also data downloads. The platform is regularly updated, so we have an update every 2 months. And as you'll see in the walkthrough later, we integrate the latest data from our providers, so it's always up to date with the best information available. And if we take another look at the knowledge cycle, this is exactly the part that I'm talking about. So we take public data, integrate it, and then enable researchers to build therapeutic hypotheses. Okay, so the Open Targets platform has a really comprehensive pipeline, which you can get a sense of in this schematic. And this is from our latest publication, which I've linked to at the bottom. So essentially, we start by looking at different sources of evidence, and we use that evidence to identify targets associated with diseases in our ontology. The evidence we collect is then scored in order to prioritize associations. And in addition to this, we have a range of annotation data, which you can use to give further insight into the targets, to refine your list of potential targets. So for example, you could look at the tractability data that we have or the safety assessments that we have. And the platform is based on a specific data model. So we really talk about 3 entities. These are targets, diseases, and drugs. So let's start by talking about targets. So we define targets as any naturally occurring molecule that can be targeted by a medicinal product. And usually these will be proteins or non-protein coding genes. We use the EBI's ensemble database as a source of human targets in the platform. We then have diseases and phenotypes. So these are both common or rare diseases and phenotypes, which we define using a modified version of the experimental factor ontology. And then finally, our third entity is drugs, which we define as any bioactive molecule with drug-like properties, and we use the Campbell database as a source of drugs in the platform. So drugs are linked to targets via their mechanism of action, and they're linked to diseases via their indication. But the true power of the platform, and the really important thing that you have to remember, is the evidence that we collect, aggregate, and analyze and use to make these connections between targets and phenotypes. And this is really worth thinking about because this is the big selling point of the platform. This is something that very few other tools do as comprehensively as we do. So what we call evidence, it's basically units of information from different sources that pinpoint a target as potentially causal for a given disease. The Open Targets pipeline retrieves this information, this evidence, from a range of data sources and standardize it. So as you can see here, we pull in evidence from more than 20 different public data sources. Now, you might be familiar with some of them, and you'll know that some of them provide maybe numeric information, some of them provide text-based information, some of them might be predictive, some of them might be reviewed. And because of this variety, our pipelines have multiple steps to aggregate, integrate, validate, and score the information in order to contextualize it and weight the underlying evidence. So for example, evidence which is reviewed by a panel of experts will carry more weight than evidence which is maybe just predicted. Once we have all this evidence, we then group our data sources into data types. So there are 7 different data types, and these are listed here. They are genetic associations, somatic mutations, drugs, pathways in system biology, text mining, RNA expression, and animal models. So to give you an idea, one of the data types is genetic associations. And within genetic associations, we'll group together multiple data sources. So for example, within genetic associations, we have evidence from our own Open Targets genetics portal, as well as the PHEWAS catalog. On the right, you can see a screenshot of what an associations page looks like. So on the left is a list of all the targets which are associated with a particular disease. These are ranked by their association score, the overall association score, which is a number between 0 and 1. And here it's represented by the shade of blue. So the darker the shade of blue, the higher the association score, the more compelling the evidence is for that association. This page also gives you an overview of the evidence that we have for each of the data types. And so you can get an idea of which has evidence which is more compelling, for example. And that's a way to narrow down the targets that you might be interested in. So as I mentioned, our platform is regularly updated. And so our most recent release was in April. And as of that release, we now feature 29,000 targets, 14,000 diseases and phenotypes, and these are linked through 11 million target disease associations and over 30 million pieces of evidence. This is huge. But essentially the main takeaway from this is that the platform really is a big data platform that integrates and analyzes a large number of data sources to build target disease associations. Okay, so I realized that I've thrown a lot of words at you, a lot of different scenarios, a lot of different pictures. And if you're like me, this will really only make sense if you see it in action. So I'm going to shift to talking about some of the use cases of the platform so you can get a sense of the different scenarios in which it might be useful. So the web interface is the first point of access for most users, and it has a fantastic new design, which is really intuitive and easy to use. And I think it demonstrates the kind of data that we have and the range of data that we have and the capabilities quite well. So I'm just going to shift to showing you the platform. Okay, so the first use case for the platform is I want to find targets that are associated with a specific disease. So let's imagine that I'm a researcher working on colorectal cancer, and I want to know what are the kind of proteins and genes that are associated with that disease and what evidence there is for those associations. So the first thing I would do is use the search bar and go to colorectal cancer. So this is the associations page, and I can see at the top that the platform has found 11,000 targets associated with this disease. And this is the screenshot that you saw earlier with all the associations and their scores. Now, I might want to refine this depending on a number of criteria. So for example, I could filter based on data types or pathways or tractability criteria. And in this case, let's say I'm interested in the targets that have genetic evidence. So I'm going to click on that, and you can see I've refined it down to 621 targets. So now I might want to explore these further. So let's go to, say, MLH1, which we can see has a score of 0.91, so quite a high ranking target. Now, if I click on MLH1, this is the evidence page. So this is all the evidence that we've collected to support the association between the target, MLH1, and the disease, colorectal cancer. One of the cool things that we have in the next generation platform is this section here, which is all our summary widgets. And essentially this gives you an overview of what kind of data we have available, which data sources have available information, and how much information they provide. So I'm looking into MLH1 and I'm thinking, I wonder what kind of mutations there are. The data source for that would be the cancer gene census. So let's look at that. So if I look at this widget here, I can see the kind of mutations that there are, the samples, the information we have on the samples. And if I click on the handy publication drawer, this links out to the literature that we have available to support this association, this evidence. We show the abstracts and the full text when they're available. So that's something that you can explore further. Okay. I've explored this association. I want to find out more about MLH1. So let me click on MLH1 here. And here you have the profile page for this target. So again, the structure is very similar. You have a little description at the top. You have the synonyms here, and then you have these summary widgets which tell you what information we have available. Now, one of the cool new features that we've integrated is molecular interactions. So through a collaboration with Intact, we have integrated information on pairwise, functional, and physical interactions of this target with other targets that we know of. So you can explore this here. We've separated it into the different sources, and you can click out and find out more about any of these associations that you see here. Another cool feature that we have is the bibliography section. So this was really a lot of work, and because we've created an entirely new pipeline. This was a collaboration with European PubMed Central, and we have a new literature mining strategy which uses natural language processing. So it uses a deep learning method for named entity recognition, and it reports sentence level matches of our three main entities, so targets, diseases, and drugs. So basically, using the algorithms, you can explore the universe of entities and how similar they are and how similarly they are talked about in the literature. So one thing you can do would be, for example, here, I want to see which targets are mentioned similarly to MLH1 in the literature. And let's say I'm interested in PMS1. This will show me all of the literature that mentions MLH1 and PMS1 in a similar way. And in that way, I might find other targets or other information that might help me with my research. Okay, that was use case number one, which was I'm looking at a specific disease and I want to find more information about the targets that are associated with that disease. Now, something else you could do would be to flip it the other way around. So let's say you're looking at a specific target and you want to know the diseases or phenotypes that that target is associated with. So for example, I could look at IL-2RA. And here you get a similar page that we had before. It's just showing you all of the diseases that we have associated with this specific target, IL-2RA. So here again, you could filter depending on what you're looking for. And one cool thing about this page is that we have different views to help you visualize the data in other ways. So for example, the graph view shows you the structure of the diseases from most general to most specific, and you can refine this based on your minimum score. We also have a bubbles view, and in this case, it's grouped by therapeutic area. So for example, I might look at, if I'm specifically interested in nervous system disease, I might look at nervous system disease here, and then I can see the different diseases that IL-2RA is associated with. So let's look at, for example, multiple sclerosis, because that's one of the highest ones within nervous system disease. If I go to the disease profile for multiple sclerosis, again, you have the profile page, which is structured the same. And in this case, I might want to look at, say, known drugs. So here we integrate evidence from Campbell, which shows you all of the drugs that we know are indicated for multiple sclerosis. And we have integrated as much information as possible from that source. So for example, you can look at the type, the mechanism of action, the action type, and you can click out to links which will either take you to the platform or to external sources for more information. Something else you might want to take a look at is clinical signs and symptoms. So here we list different phenotypes that are associated with multiple sclerosis, and this might help you find other targets that are useful because they're associated with these specific phenotypes. Okay. So that was use case number 2. I'm looking for diseases associated with a specific target. But if you remember our data model, I told you that we have 3 entities. So targets, diseases, and drugs. So the last thing you can do is look for annotation information on a specific drug that you're interested in. So let's say, for example, I've heard about this drug, metformin, and I want to find out more about it. I want to know, is it authorized? Is it approved? Does it have any warnings? What else do I know about it? So if I get to this page, the metformin profile page, I can see at the top, well, it's been approved, but it has some warnings. So now if I go down to the warning section, this is something that was updated in the latest release of Kemble, so new data that we have in the platform. These are the black box warnings. These are the most severe warnings that the FDA gives out for drugs. And you can see that metformin here has a number of different warnings associated with it. If you scroll down, we have another kind of information which we get from the FDA, which is from the adverse events reporting database. In this case, we integrate reports from healthcare professionals of events that have happened after metformin was prescribed. And these are some things that you can look at here, for example. Okay, so that was everything I wanted to show you for the walkthrough of the platform. If you have any questions, you can also ask them in the chat, and I'm happy to go through more uses later. So now that you have an idea of what you can look for in the platform, let's talk about how you can access the data. So there are a number of ways in which you can access, explore, analyze our data, depending on how complex your question is, and also how programmatically savvy you are. So I mentioned the web interface is how most people use the platform. And using the search function, you can quickly navigate to entity pages, associations pages, or evidence pages. The data tables and visualizations you might have seen, but they're available to download in multiple formats depending on what you're looking for so that you can save, explore, and analyze them. But the web interface is really only suitable if you're looking at a single target or single disease or single target disease association. It can't really handle anything more complex than that. Another way that you can access our data is through the API. And so with the next generation platform, we are now using a GraphQL API, which is really cool because it allows you to access the data in a language agnostic manner. It also allows you to construct tailored queries so you can find exactly what you're looking for. And if you use the API playground that we have a screenshot of here, it has built-in documentation and a built-in schema. So it's really easy to get started, and you can figure out exactly how to build your query to get the information that you're looking for. However, like the interface, the API really only supports queries for a single entity or a single association. Any queries that you have that are more systematic, more complex, involving multiple targets, you'll need to use one of our other access points. So the next one that you can use is BigQuery, so Google BigQuery. So for those of you who don't know, BigQuery is a data warehouse that enables researchers to run super fast asynchronous SQL queries using Google Cloud's infrastructure. Once you've run a query, you can export the results or copy them into Google Cloud buckets for your own further downstream analyses. And then finally, the next way that you can access our data, and the most powerful one really, is that we provide downloads of all data files generated by our ETL pipelines. So compared with the API, with data downloads, you can explore specific data sets and integrate them into your own pipelines. They contain all information on targets, diseases, and drugs, relevant annotation, and we have more than 20 available for download on our downloads page. So the platform code and platform data, as I mentioned, are all open source and open access and freely available for academic and commercial use. However, we do ask that if you use our code or our data, you cite our latest publication, which I've linked to here at the bottom. You can find further details on the platform in our documentation, which also has detailed instructions and example code for how you might be able to access the data. And you can also join the newly created Open Targets community, which has users, and you can discuss with different people, get ideas on how to use the platform, and also chat to the team directly so you can get more specific answers to your questions. I highly recommend you join this community. So to recap, Open Targets is an industry academia partnership that supports research for systematic identification and prioritization of targets. Our flashy flagship informatics product is the Open Targets platform, which is an open source, freely available tool that aggregates and integrates over 20 different data sources to help you with your research. So if you use the platform, you can search for target disease associations, view the underlying evidence supporting that association, and view all the annotation data we have on these different entities. Thank you very much for listening to me. I've included here some links to our website where you can find out more information, our blog, where we feature stories from open targets and deeper information about how and why we made certain decisions, and also our socials so you can follow us and get the latest news from open targets. Andrew and I will now.
One question, and this comes from David around how the open targets is used by scientists. And so in this case, I'll talk a little bit about the experimental work that we have that is being published, and then also the informatics tools as well. So on the experimental side, a lot of our projects were sort of started about 3 or 4 years ago, and they're now beginning to publish. So we had a couple of sort of flagship papers that were published last year. One of them being on Project SCORE, which was a CRISPR synthetic lethality knockout paper by Behan et al. That was published in 2019. We also had another paper that came out around the single cell exploration of the lung. And then we've also had some other research that's come out around cytokines as well, and sort of on the inflammation and immunology space. So we have a lot of research that comes out there, and we make that research open and available as soon as it's published, and really support global research and use of our data in many different ways. And in some cases, these projects will build their own specific web portal so for example, Project SCORE built their own website to show all of the data that they were generating. They also submitted some of the data to us to integrate into our platform as well. So it really depends on, for the informatics side the experimental side, for the data to be integrated into some of our informatics tools. If it fits our data model of target disease evidence, then we can also make it available through our own channels through the platform as well. On the informatics side, the tools are used in a number of different ways. We have obviously the public platform that is available that you can visit at platform.opentargets.org. A lot of research scientists use that to identify targets and look for potential insights, novel insights based on our scoring algorithms and all the data that we integrate in. We also know that it's used in a number of other contexts. So if people are looking, for example, for specific information about a given target, a given disease, or a given drug, which Helena showed how you can search for that. So our informatics tools really support a number of different use cases. All of the data, similar to the experimental side, all of the data is open source, open access. We are a big believer in open science and open data. So you can take the data and you can use it for either academic or commercial purposes as well. So we have a lot of users, and we're hoping that through this webinar and through continued sort of outreach, that we're going to get more users that are interested in using all the different types of data that Open Targets generates to support their research. So then there was another question around how the score is calculated. I will just, without getting into too much detail here, I will say that the best thing that you can do to learn about the score is to visit our documentation, which is at platform-docs.opentargets.org. And I'll put that URL in the Q&A. And the reason why I say that is we've got 2 pages there. One page that lists how we score each particular evidence that comes in from our data providers, and then how we generate the overall association score, because we actually have 2 different steps. We score the evidence individually, based on our review, our data team, and our expert review on how the piece of evidence should be scored. And then we aggregate that information all together using a harmonic sum algorithm to then generate these overall association scores between targets and diseases. Both of those bits of information are available in our documentation, so I'll make sure to include that as well. Okay, we have another question here around how often data is updated in the platform. Helena, do you want to take this question?
Yes, sorry, trying to juggle answering questions and looking at what's coming in. Yes, so Data is updated every 2 months. So we have, well, roughly, so we have 5 releases a year. If you keep an eye on the blog or on our social media, we also have a newsletter. You can find out when new releases are coming out. You can also join community. There are lots of ways to stay in touch, but if you join community as well, that'll tell you when new releases are coming out. So 5 times a year is when we update, to answer your question.
Perfect. Yes. And there was a further follow-up question around the scores changing. Yes. Every time there's an update. Yes, sometimes the scores do change as we get more information in. A lot of our pipelines are trying to pull the most late-breaking information. So, for example, every time we do a release, we always pull the latest clinical trial information. Our literature pipeline is up to date as of maybe 2 to 3 weeks before we release. So we really are trying to provide as late-breaking information as possible. So there is the chance that, yes, the score might change. For example, if there's a new drug that goes into a clinic that targets a gene that had never previously been targeted, that will absolutely adjust the overall score, because we do have a lot of confidence in the Campbell evidence. It does get scored quite high depending on the phase of the clinical trial. So there will be some changes every time that we do an update. We recognize that when you're doing research, particularly if you're looking to use our data to, let's say, validate a model or to test another model or integrate and interrogate the data more, you want a specific sort of snapshot of the data. So because we make all of the data available for download, you can very easily go back to the files that we generated, whether it's 2 months ago or 2 years ago, and you can use that data and that historical data to also chart and see, you know, for example, how a score might have changed, or to see what the scores, let's say, overall would have been before we made some major changes. So, for example, a few years ago, this is probably about 2 years ago, actually, we changed from EFO2 to EFO3. So our ontology expanded significantly because the EFO team at EBI had done a fantastic job of reorganizing the ontology and making sure that we had better representation across a number of different therapeutic areas. In doing that, because we propagate our evidence through the ontology, it did cause some scores to change, as now some diseases that might have been in one therapeutic area are now in another one, and some of the higher level diseases or therapeutic area terms, those scores changed. So we know that those are big changes that come in. Helena does a fantastic job about communicating all of those changes when they come out through our blog posts and through our social media and our community. So you will know about what those changes are and why some of the scores will have changed. But again, because we make the data available, we do try to ensure that you can still go back and look at that data and compare it if you were looking for a very specific snapshot as well. Okay, I see that there's another question here, and I apologize, I might be jumping around a bit, so I'm going to try and answer as many as we can. Can we use a system for other diseases such as diabetes, fibrosis, or is it focused only on cancer and inflammation? We actually cover all disease areas and therapeutic areas that are found in EFO. This is actually, I will say, an interesting conversation that Helen and I had earlier, because we were trying to figure out how we should define systematic. And we really, as a consortium, we define systematic as being across a number of different therapeutic areas. While our experimental research is focused in oncology and inflammation, immunology, and neurodegeneration, our informatics tools are very much so disease or therapeutic area agnostic, and are really looking systematically across a number of different diseases and therapeutic areas. So we know that people, for example, will use our tools to look at, as you say, diseases and phenotypes like diabetes and fibrosis, and not just cancer and inflammation or immunology branch disease terms.
I'll just add as well, I remembered when you were talking about the association score, I should point out, so the association score isn't a confidence score. Just because the score is really high doesn't necessarily correlate to how confident we are that that target really is associated with disease. So that the score will be skewed by how much evidence is available and the quality of the evidence. So I should have mentioned that, but yeah, it's not a confidence score. It just reflects the evidence that we have available.
Yes, very good point. So we had a couple of questions around pathogen genes and proteins. So as Helena mentioned, we generate our gene index, if you will, from the Ensemble Human Gene List. That being said, within some of the data, you will find information around, for example, viral proteins. So, for example, if you were to go to the ACE2 profile page, the molecular interaction feature that Helena provided a bit of a spotlight on does include information about how that particular gene interacts with the SARS-CoV-2 viral protein. So there is information that we do bring in around viral proteins and pathogens, but in terms of searching, it would be searching based on a human gene that is found in Ensemble.
Yeah, linked to that, there's also a question about if we can use this platform for COVID-19 studies. So you can't use the Open Targets platform, but when COVID-19 started, we did create a separate portal for COVID-19. So it's covid19.opentargets.org, and you can use that to explore the different information that we have for COVID-19. So that integrates data on both human proteins and COVID-19 proteins.
Perfect. And then we have a question here about how to validate targets by using an in silico approach. So at the moment, we actually have a validation lab that is working on some validation experiments that are coming out of our experimental side. So looking at taking some of those targets to generate some further insights there. In terms of the validating of targets, what we really do is provide a breadth of information that you can have access to different data points to really look at all the information that might be available about a specific target or disease that you can then integrate in to make decisions. And the reason why we do that and we don't sort of provide this direct validation is that we know very clearly from a lot of our users that they might be looking at different information in terms of sort of validating a target. And what I mean by that is we know that we have users that will visit the platform because they're looking for drug repurposing opportunities. And so for them, the concept of validation might be a little bit different because they're looking at a target that might already have an approved drug and maybe looking at some evidence for a different indication. So for them, the validation might be slightly different than someone who's looking for a novel target, has never been in clinic, and they're looking to develop a drug there. So we try to provide a breadth of information, not only about the evidence supporting the target disease association, but also for the respective entities, so that then as a user, you can sort of make that decision around how you choose to validate that information as well. And sorry, give me one moment here. There's a question around the strategy for novel data sets or naive gene proteins. Could I ask that user to maybe just type in a little bit more information just so that I can try to answer that a little bit more effectively? Helena, there was a question around the text mining. Could you?
Yes, sorry, struggling to unmute. Problem of 2020.
Yes.
Yes, so the text mining. So it's a new pipeline that we have in the next generation platform. We used to have a different way of looking at it, but we now have this pipeline which we've developed, which uses natural language processing. So it uses a deep learning method for named entity recognition. And what it does is it looks through the sentences and it identifies co-occurrences of the entities that we have in the platform. So the drugs, targets, or diseases, and reports those within the widget that we have. So when you can filter depending on if you're looking for targets or diseases or drugs, and it'll suggest other entities that are mentioned in a similar way to the one that you're currently looking at? I'm not sure if that answers your question, or Andrew, if you have anything else that you want to add.
No, other than just because there was a similar question around the actual NLP model as well. So I have included there a link to the documentation. So again, platform-docs.opentargets.org has a fantastic page called Bibliography that actually explains this approach. It was written by our technical team. So it really provides some insight into the Word2Vec model that they developed and validated, and how they've been working with Europe PMC to really provide, I think, a new approach to text mining and really to navigating, I think, a knowledge graph of literature, which we know is a big ask for a lot of researchers. And I will also just say on that as well, just to really reemphasize a point that Helena made earlier, also around the fact that a lot of our evidence is backed by a publication as well. So with the PMID or Public ID that we get from our evidence providers, we also call on Europe PMC just to provide a little bit more information about that. And we do that specifically. We know that in some cases, there's a lot of ongoing research, let's say, around a particular target and disease association. And so by providing information about the citation, maybe when it was published, where it was published in the abstract, it provides a little bit more insight that you as a user can use and take advantage of when deciding if this is a publication that you'd like to read further or explore further, because it might be relevant to your work as well.
There are a couple of questions linked to that. So is that only on open access full texts? And was the NLP performed on just abstracts or the whole paper? I'm not sure, Andrew?
So yes, so at the moment, the NLP was done on the abstract. Because using Europe PMC, the entire sort of corpus of literature, it was done using that approach initially for this initial one. There are further iterations that we are planning on looking at, and this is so that we can, I think, continue to really make sure that the information that we provide and the insights that are generated are very useful. So we are looking for any feedback, particularly through the community, on the approach that was taken, because we really want to make this as robust as possible, not only for, as I say, and as Helena said, the fact that we have the platform that you can use at any point, but also because you can take the pipeline, you can take the data sets as well. So we would definitely encourage you to really have a play with it. And I will say that the engineer, our technical lead that worked on this, Miguel, is very keen to hear from users who have really interrogated the literature data, because this is sort of a pet project of his to make it really robust, because we think that this is something that could be a really big game changer for a lot of different use cases. So there was a question here around collaboration with hospitals for data collection. So at the moment, we don't have patient information in the platform. We are actually looking at building in more information about patient stratification through different threads in the platform. We've started to get to grips with this. So in the disease or the phenotype profile page, we now have a section called clinical signs and symptoms, which was data that has been pulled in from a number of different sources, including EFO, HPO, and Mondo. And in looking at sort of how we can present more information about a specific disease or phenotype, one of the things that have come up is sort of more information about patient stratification and information that might be available. We also, though, have another way of getting in some information from patients, primarily through the FDA adverse events reporting data. So as Helena pointed out on the metformin profile page, we do have the adverse events data that we do integrate in. And that data, to provide a higher confidence, we only screen in, or we only integrate in the data that's been reported by a healthcare professional so as to make sure that it's as of high quality as possible. So that will also include some information that will be coming in from hospitals as well. There is another question here around correlating score and prioritization of targets, which, as Helena mentioned, the score is one that we have generated, but it's not necessarily predictive. It's based on us integrating a wealth of information to provide some insights there. We know that a number of papers have actually been published around using the open targets data and comparing our algorithm and our approach against other benchmarks. And I think it depends on how you weight some of the data as well. I think in terms of the prioritization, I will just also say another thing that's important here is that the score really has to do with the identification of the targets. So for example, you're looking at a given disease, let's say colorectal cancer, and you're looking at all of the targets there. So we're using the score to help you identify and to do a preliminary prioritization. But for us, we're really looking also at building out a lot more entity annotation to support prioritization. So for example, tractability. Has the target ever been in clinic? Is it known to be potentially tractable by an antibody, a small molecule, or another kind of drug? Is there any safety concerns? What is the baseline RNA and protein expression looking like? So for us, the reason why it's difficult for me to answer this question is because in many respects, the score is really around how we help you identify the larger number of targets that might be associated with a specific disease, or the flip way, the number of diseases that might be associated with a target. But then the prioritization, I think, really comes in with a lot of the very specific entity annotation, as I say, tractability, safety, baseline expression, etc., that I think really will help a user more fine-grained, will generate a more fine-grained ranking of potential targets, based on the fact that it might be potentially tractable, there aren't any known safety issues, it's expressed in the right tissue, and for example, there's a mouse phenotype that's available that I can look at to investigate a little bit further as well.
So there's just a question that came up in the chat about citing the platform data. So I'm just going to answer it here. So it's in the slides. So those will be available afterwards for you to look at. And it's also on the documentation, the full citation of the latest paper, which is in nucleic acids research. So I can't remember the ID off the bat, so I'm not gonna give it out. But if you go to the documentation, you'll find it there.
Perfect. And I'm just trying to see here if there were any questions that we missed. I did mention already about the in silico validation, as I say, is really using all of the information that we have and allowing you as a user to decide what information would be important to validate. And I guess just on there was that question about sort of novel data sets and genes and proteins. I will also say that one of the things that we also do in terms of supporting maybe more novel data sets coming in is that you can create your own instance of the platform. So as Helena said, all of our code base, all of our data is open access, it's open source, it's open data, open science. And so if you've got a specific data set, maybe that's internal, that you don't want publicly available, or it's not publicly available, but it does fit within this model of target disease evidence, you can very easily create your own instance of the platform. You can spin it up, you can have it running. We know that a number of pharma companies, I won't say which ones in case any of them are on the line, but we do know that a number of pharma companies do have an instance of the platform running internally. Specifically, I think to deal with some of these more novel data sets or unique data sets that they have that either are based on experiments that they've run or analysis that they've done that are not publicly available. So there are ways of either taking the data that we make available in all the different formats that Helena said, and you can integrate that into a pipeline in any which way, or you can also take all the code base, you can spin up your own instance of the platform, you can inject in your own data and run the computational pipeline against all of your data, and it will score your data and it'll present it to you within the UI as well. So you can make use of that as well. And I believe that is it. There was one question around pathway identification in a medicinal plant. We don't have any plant-based information in the platform, but there are pathway. So we do integrate pathway evidence in when you're looking at target disease association. So we generate that data and scores from Reactome, as well as some manual curation that's been done by our team as well. And likewise, on the target profile page, you can also find information about the pathways that a given target is found in as well, so that if you're looking for specific pathway mentions, you can also do that as well. And again, this is all the information that will be available in different ways, available for download and access, so you can make use of however complex the data that you want. As I say, if you just want to know this gene is in this and this and this pathway, we can give you that. But if you also want to know about some of the evidence that maybe connects this gene to this particular disease with this pathway evidence, we can also provide you with that all the way down to, I believe, the reaction. We provide the reaction and reaction identifier as well. So not only the parent pathway, but also the actual reaction itself.